Hi everyone, my name is Erick Navarro. I'm a PhD candidate in bioinformatics at the University of British Columbia. And I'm very happy to be here to present some of my work that is related to the development of this R package called RAMEN that aims to untangle gene environment contributions to DNA methylation variability. So I will first start talking about what is DNA methylation. So DNA methylation can be defined as the addition of a methyl chemical group to the DNA, specifically in the cytosine, in a CpG context, which means a cytosine followed by a guanine in vertebrates. DNA methylation is involved in many biological processes, and most of these biological processes affect gene regulation. In one of the most studied examples is when DNA methylation happens to be in the promoters, for example. And in this case, when DNA methylation is present in these promoters, this leads to an inactivation of the gene. And therefore, this can be associated with phenotypic variation in some cases. So overall, studying DNA methylation variation is important because we can understand the sources and the dynamics of this DNA methylation variation. We can understand some of the underpinnings of phenotypic variation in humans. So I've talked about DNA methylation variation, but what are the sources, right? So the two main sources of DNA methylation variation are first the environment and then genetics. There are many papers that have associated how differences in environmental exposure, such as, for example, smoking, can lead to changes in specific sites of DNA methylation, and same for genetics. However, we don't currently have a very good understanding and grasp on how widely the environment acts on DNA methylation, and the same for genetics in a global context, and also if these two factors act mainly individually or jointly, and if it's jointly, if it's through additive or interaction effects. The difference between additive and interaction effects is that with the additive models, this means that the effect of the environment and genetics on DNA methylation variation is independent. In contrast with interaction, in the interaction models, the effect of one environmental exposure on the DNA methylation would depend on the genetic makeup of the individual. So you can think about this like a genetic susceptibility scenario. So to understand this question and answer it, I have developed a package called RAMEN that stands for Regional Association of Methylome Variability with Exposome and Genome. This R package contains 6 functions that conduct the gene environment contribution analysis to DNA methylation variability and integrate matched microarray methylome data, genomic variants, and exposome data from human cohorts. Because of the high dimensionality of this data, these functions are compatible with parallel computing, so they run very fast. So I will talk about briefly how RAMEN works, and I will also highlight some of the key methodological implementation that we have here. So briefly, RAMEN can be separated in 3 main steps. The first one is to identify highly variable probes, or which means just out of all your data, you can identify the sites that have high variability of DNA methylation. After that, we cluster them in what we call variable methylated regions, or VMRs. The reasoning for this is that we know that CpGs that are close together usually have a very similar epigenetic profile. So if we were not to cluster these in regions, we would be testing redundantly several sites that have the same information. So we do this to increase our statistical power and also reduce the computational complexity of the models. And finally, once that we identify these VMRs, for each of them, we model the contribution of the genome and the exposome. So in terms of the methodological highlights that I want to talk to you today, first is the package development. This is the first R package that specializes on this type of analysis. And in contrast with previous methodologies, we have also modified the definition of the VMRs. So the canonical definition of VMRs are different CpGs that are close together and that share a similar epigenetic profile, which are these ones. So what is the problem with this definition? The problem is that this assumes that you have a very good coverage of your data, which could be true for some technologies, such as, for example, whole genome bisulfite sequencing. However, in the human world of DNA methylation, this is very expensive, and if you want to apply this technology to a big data set, this is not very cost effective. So for human populations and epidemiological cohorts, what is used the most is DNA methylation microarrays. And as we know, these microarrays have a very specific design that target probes that had been curated previously. And for instance, most of the arrays, to maximize the information that can be extracted from these arrays, they, by design, set the probes in positions that aim to recover the DNA methylation state of the whole region. So for instance, the EPIC array, which is one of the most used arrays in DNA methylation, most of the sites that cover DNA regulatory regions happen to be represented by a single probe. That again, by design, it is positioned in a place that represents very good the region. So by using only the canonical VMR definition that has been used before in previous approaches, this removes a lot of information that is very valuable and that is present in the arrays. So to do that, to address that, we have also implemented a new category of VMRs called non-canonical that incorporates the information and the design of the arrays so that we can also recover this information and model the DNA methylation in these places. Another highlight that I want to mention is that, as you can imagine, we are working with a lot of SNPs and a lot of environmental variables and also a lot of sites. So this leads to very complex and big models. So we have implemented a machine learning-based method that what it does is that prior to the modeling and comparison of all the models, it first for each VMR screens all of the variables that we have in dataset so that we can select only the ones that are potentially relevant, therefore reducing our computational time and also increasing the performance of the methods. After that, we feed the models and we select the best one. And the final highlight that I want to mention is that we have also implemented a step to, after identifying the model that best explains the DNA methylation in each of these places, we want to know that the model that we're selecting is not just a good model out of very bad models, right? So to do that, we have implemented a methodology based on permutation that allows you to simulate a null distribution so that we can identify and remove the models that have performance that is not better than chance. So that is about the methodology. So I want to show you some of the results of how this looks in action. So I will show you results that we obtained analyzing the child cohort, which is a Canadian nationwide longitudinal cohort that recovers information from families all over the country. So from this cohort, we used 699 cord blood samples that after the QC, we had information about the DNA methylation state of around 800,000 CpGs, also around 1.3 million SNPs, and also this is really important and rare, a very deep prenatal environmental characterization of 94 environmental variables. So these samples were taken at birth, and I didn't have time to go into the biology of this period, but the prenatal stage is a really relevant stage in human development that is very sensitive. And it is heavily studied under the developmental origins of health and disease framework that states that during this stage, specific alterations can lead to long-term physiological and molecular alterations that can lead to disease. So we're working here at birth. In terms of the prenatal environmental characterization, we have information about the parental psychosocial, which includes depression, stress, etc. of the parents, also parental health, which are health conditions during and prior to pregnancy, also nutrition about how the mother ate, and also built environment, which is related to exposure to pollutants of the fetus. So what we observed is that when we feed and compare the models, again, just a reminder, we feed 4 different models. One of them is the DNA methylation of one genomic region being better explained only by genetics, only by the environment, by an additive effect of both of them, or an interaction. And we also have a non-conclusive category, which, as I mentioned before, is all of these models that we didn't have a good performance enough to be confident enough to have a result there. And all of these models were adjusted for sex, age, cell type, estimated proportions, and genetic ancestry. So what we observed here is that genetics was present in most of the winner models, usually in combination with the environment, which is here mainly in an additive fashion and then interactive fashion. This is really interesting because historically DNA methylation has been thought to be mainly impacted by the environment. And as we can see here, the environment alone almost never explains the DNA methylation state of these regions. Something that RAMEN does after identifying the best model is that it also conducts a variance decomposition analysis so that we can estimate how much of the variance is coming from each of the factors that we're modeling. And when we analyze these results, we can see that across all the scenarios where the genotype is involved, the SNP term usually explains a way higher amount of variance compared to the interaction and the environmental term. And finally, we wanted to compare our results with an independent cohort. So we collaborated with Dr. Darina Samara, which is a collaborator in Germany that had access to a cohort called PRIDO that is Finnish and that also has DNA methylation, genotype, and environmental information of children in cord blood. And what we observed is that in the overlapping VMRs, all of the effect of the genetics was 100% consistent in all of them. So as take-home messages, RAMEN provides a reproducible pipeline to conduct gene environment contribution analysis. Also, I didn't talk too much about this, but RAMEN also, all of the functions that RAMEN has have independent behavior, so that they can be very useful for other type of analysis. For example, the main application could be to reduce DNA methylation data dimensionality. And finally, we also found that genetic variation plays a key role in DNA methylation variability at the prenatal stage, usually in combination with the environment. Thank you so much for your attention, and I'm open for questions.
Hi, in your conclusion, you saw that the environment has got not a very strong contribution. No?
Mm-hmm.
And do you think it's something to do with the fluctuation or the variability or the not precise? Because you are measuring the environment with these 8 variables. What variables?
Oh, so this is for the comparison,
Yeah,
but for child, this is 94 environmental variables. So it is these ones. So these are just some examples, but they fall in these 4 dimensions that are parental psychosocial. So here we have stress, depression, socioeconomic status, the social dynamic between the chronic stress events of the mother. All this is on the maternal part because it's kind of conceptualized as the environment of the fetus during gestation. We also have parental health of the mother as well, nutrition, and built environment. So there are many hypotheses that I have on why the environment in this stage seems to be playing a very small role. One of them is that biologically, the fetus is protected by different extraembryonic layers that aim to protect the baby from all of these environmental exposures. So it could be that the physiology of the mother kind of modulates all of these environmental exposures. Another could be technical problems, such as we are, in contrast to the genetics, for genetics, we have protocols that aim to very optimally recover genetic variation. For the environment, we don't have that. And also the environment is dynamic in contrast with the genotype. So we had like these variables for 3 time points before birth, but it could be that we didn't recover like the exact moment where this environmental exposure was relevant or not. And in addition to that, we also have noise with these, like a lot of these variables were we collected with questionnaires. So it's really, really hard. Like the whole field of studying and integrating the exposome is really hard and has a lot of biological and technical challenges that could be behind this. Yeah.
Thank you.